So we have already discussed the first part of meiosis 1, that is prophase 1. In detail, we have discussed the 5 substages of prophase 1, that is leptotene, zygotene, pachytene, diplotene and diakinesis. At the end of prophase 1, as you can see here, the chromosomes are getting ready to be aligned into the equatorial plate, and the spindle has also quite been established by these centrioles which are moving away from each other. What happens in metaphase 2? Metaphase 1? How is metaphase 1 different from metaphase of mitosis? In metaphase of mitosis, chromosomes formed a single line, but notice here the chromosomes are forming a double line. They come together in the equator along with their corresponding pairs. Even when they come and align themselves in the equatorial plate, they are still forming a pair. They are still a part of a pair. So they come as homologous pairs in the equatorial region. So we call this as the double metaphase plate. And now in anaphase 1, a process called segregation or disjunction takes place, where the pairs are getting pulled apart from each other. What were once a pair are now becoming single. So pairs become single as they get dragged apart. Notice that their centromere does not split. In anaphase of mitosis, their centromere splits, but here the centromere is very much intact, and each chromosome has two sister chromatids attached to the centromere or attached at the centromere. So there is no question of centromere splitting over here, but as a whole the chromosome is dragged away from its corresponding partner or its corresponding pair. And then during telophase, the chromosomes which have now become single reach the opposite poles, and of course, as you can see here, cytokinesis sets in the form of cleavage furrow, and you end up with two haploid daughter cells. If you noticed, the cell that we began with had 4 chromosomes, but now each of these cells have 2 chromosomes, and therefore we say the number of chromosomes has been reduced by half, and hence we call meiosis 1 as reductional division. Now both these cells have become haploid with 2 chromosomes each. Now they step into meiosis 2. Each one of them step into meiosis 2. So notice that if I call this cell as cell A and if I call this cell as cell B, A is also stepping into meiosis 2 and B is also stepping into meiosis 2, and meiosis 2 is very much like mitosis. Notice how during prophase the nuclear envelope is disintegrating, and we don't just call it as prophase. Please, you have to affix 2 always. Prophase 2, metaphase 2, anaphase 2. Only then we would know that we are talking about meiosis and not mitosis. So during prophase 2, the chromosomes become visible, and you can see how the spindle apparatus is getting ready. During metaphase 2, notice how the chromosomes are aligning at the equator in a single line, not a double line. Double line was in metaphase 1. This is metaphase 2, which is very similar to mitotic metaphase. The two chromosomes align one beside the other, that is in a single line, not in a double line. And then, very important, the centromere of the chromosome splits. If you remember, I had told you that the chromosome has a centromere and there are two sister chromatids attached at the centromere. So the centromere will split. The sister chromatid above will get pulled to the upper pole, the sister chromatid below will get pulled towards the lower pole, and that is what is happening during anaphase 2, exactly like the anaphase of mitosis. And then the chromosomes that got the sister chromatids, which now become chromosomes that get pulled towards the opposite poles, they reach the opposite ends in telophase 2, and you can see that there is reformation of the nuclear envelope, and of course again the cytokinesis sets in, and here we have 2 daughter cells and here we have 2 daughter cells. So totally we have 4 daughter cells that are formed by the end of meiosis, and if you notice, each of the daughter cell has only 2 chromosomes, and that is what is the significance of meiosis. Because meiosis occurs in 2 steps, that is meiosis 1 and meiosis 2, by the end of meiosis 2, we have a total of 4 haploid daughter cells.